would put this on a seal in this diving hole and then recover it. And it was the first time we were able to get a time and depth record, but it was only good for about an hour. So when I joined his lab as a postdoc, I'd finished my work here. I'd worked on sea otters. I really wasn't doing that much on diving behavior. Jerry Koyman said, we got to do more than this. And so this is was talk about a strip chart recorder. Another thing that graduate students say, when you say strip chart recorder, what's that? Well, this was a mechanical system where you had the film here. The film would then roll across this thing and then be taken up in a spool here. Now there's a light emitting diode at the end of this arm. And in this tube here is a Bourdon tube, which as the animal dives, that Bourdon tube would untwist and that little arm would go across the film and then it would record an image. And then here's a picture of it when we put this out on an Antarctic fur seal. Now, what people complain today about, gee, their very cool instrument didn't work. These things we had to load in the dark. You can see all these really nice little electronics because it's film. We'd go in the dark room, load this thing up, hope we didn't pinch the O-ring, get the batteries connected, and then put it all together and then put on a seal. It would last 2 weeks at the longest. And then bring it back and you had to recapture the seal and then hope that the instrument worked. And then you have to unload in the dark and then develop the film in a dark room. And then hopefully you got a record. The other thing that's interesting is this film was super, super thin. And why was it super thin? Well, we wanted to roll as much of it as possible on the spool. And when we called Eastman Kodak to get it, they said, there's only one other organization, they use the word company, one other organization that buys this film, but we can't tell you who it is. You might guess who that might have been. It was the defense establishment, because back then they used the super thin film in the Corona satellite system, or the keyhole satellites, which were the way that they would have a satellite which would fly over, take pictures on film, and then jettison that film that's in a cartridge, and the Air Force would fly and capture the thing as it parachuted down. I now know that because all that stuff has been declassified. But so we were using the same technology as the state-of-the-art defense industry. This is what it looked like. I was fortunate enough to make my first trip to the Antarctic working with Jerry Koyman. And you can see that time depth recorder now placed on the back of a Weddell seal. And this was the first time we were able to do this with a Weddell seal. As I said, we developed that film, and then we make this really long strip chart. It was a flowzerography. I don't completely understand, but we enlarged that little piece of film, and you get this piece of paper, and then you sit there and hand digitize this thing. So one record would take about literally 24 hours to hand digitize on a big digitizing board. And then we'd figure out how deep and how long. We'd also roll the things down the hallway and then just make up stories. Well, gee, what do you think the animal is doing here? It dove for 5 minutes here, but now it dove for 20 minutes. And over here, it stopped diving. And it was really, this was all brand new stuff, and we had no idea what these animals did. The other aspect of this is we didn't know where they were going. When we put them out on elephant seals, we knew the animals left the beach, and then we'd wait for them to come back, and then we'd recover the dive recorder. When they were diving to 600 meters, we knew they were diving out in the open ocean, but that was as much as we knew. So this is that time depth recorder, all in 1983, 84 on an Antarctic fur seal. This is much more recent. This was a picture taken in 1998 on the same species. And now you can see the difference in the picture on the left. That's that photomechanical time depth recorder, and the picture on the right, that little yellow circle, shows now an electronic version of that digitally takes pressure readings every 5 seconds, and then you pop it into your computer, download it, voila, you've got data. We also had little radio tags that tell us when the animal came back on the beach. But now the other big change is we could follow these animals anywhere on the planet using a satellite transmitter. So the little circle here now is a satellite tag that would tell us where the animal was. So now we have a combination of diving behavior and depth, diving behavior depth, and where the animal is actually performing those behaviors. The technology continued to develop, and this was 1997, and this state-of-the-art was 512K of memory, and we were really excited to get a whopping 512K of memory. This device has been around since 1997. It's kind of the workhorse, and this particular version has 16 gigabytes, but that's memory in terms of these devices is now not really that big an issue. That little device is about that long. The other change is they're now potted in resin. If you look at this one, that's a titanium housing that was machined, and a lot of the cost of this tag was making the housing. And so when you just pot them in resin, it makes it a lot less expensive. So what did we find? Well, this was a record now. I'm using our more recent digital recording instruments. You can see these are days, so this would be what, August 25th to the 29th. So it's a 4-day record. This dark area is nighttime, the white area is daytime. So you can see a very pronounced difference between diving at night and diving at daytime. We know that there's what's called the deep scattering layer. That's all these fish that during the daytime they stay down deep, and at nighttime they come up to the surface. So the elephant seals are tracking their prey. The other thing that you can notice is that on average, they're diving in 456 meters. It's a little shy, it's about 1,500 feet. In the daytime, it's about 619 meters. That's almost 2,000 feet. And to give you a perspective, that's about the average, the maximum dive is just over a mile. So you can imagine on a single breath hold diving down in the ocean to a mile depth. Elephant seals do that quite often. Another way of looking at it, these are the tallest buildings in the world. That's the one in Dubai. And this is what an elephant seal does. So I'll put it in perspective. These are the tallest buildings in the world, and an elephant seal is almost twice that. So if you've ever, you go to the Empire State Building, you look at those buildings, you can imagine an elephant seal can dive at least twice that distance. Pretty amazing. As this technology has evolved, other people have been able to use these devices. And what we now know is that elephant seals, which you see the diving record here, this is 16 hours. You've seen the typical diving pattern elephant seals. This is a sperm whale, which is this one here. These upper two are beaked whales. These upper two here are beaked whales. That's what a beaked whale male looks like. Beaked whales are these very secretive animals that are out in the deep ocean. We didn't used to know much of anything about them, but once they got implicated in responding to naval activities, a tremendous amount of research has gone into them. And now we almost know more about beaked whales than any other cetacean, just because of the interest. But the point being is that the deepest diving mammals are sperm whales and beaked whales. You can see that they're diving quite deep routinely, but elephant seals certainly almost match them in terms of depth. And elephant seals are the deepest diving seal or sea lion, and one of the deepest diving pinnipeds. So I said when we started, we didn't know where these animals went. This is, if you go to one of the early textbooks, this is what it said for the distribution of northern elephant seals. We know that they breed on these islands, and you can go and count the animals on those islands. And one of my colleagues that would do these aerial surveys and boat-based surveys, that's what this shows you, that these are boat-based and aerial-based surveys on the left. He said, it's really weird. We count X number of elephant seals on the beach, but when we fly over the water, we don't see them. And he says, well, either they're going somewhere else or they're spending a lot of time underwater. Well, the answer to the question is both. When you actually allow the animal to tell you where it goes by putting a satellite tracking device on it, you find out that elephant seals basically use the whole northeastern Pacific Ocean, and they only spend about 20% of the time at the surface. So if you're using a plane or a boat, you're going to really undercount them because they're not in the area where we looked. They're way offshore, plus they're spending most of the time underwater. But one of the ways I sort of think about this is we've heard, I don't know if you've heard that joke about the guy that lost his keys in the parking lot. And he's looking around and somebody says, well, where did you lose them? He said, over there. And he goes, well, why are you looking here? He says, well, this is the only place there's light. And that's my analogy with, we use the technologies we have, and they may or may not be appropriate to answer the question in front of us. So to give you a little bit better idea, each one of these is an individual elephant seal. When that little wiggle, that little worm is longer, they're moving faster. When it gets smaller, it means they're moving slower. This is multiple years in the life of elephant seals. These are females. And you can see they make 2 trips a year, one immediately after the breeding season. They come back to molt, and then they make a longer trip, which is called a post-molt trip. Then they come back the following year and then breed again. One of the great things about being here at UC Santa Cruz, other than it's a great place to be, is 20 miles north of us is Año Nuevo Reserve, which is where the elephant seals come back. That's where these animals are going to and from. So it makes it incredibly easy for us to research these animals, to engage our undergraduates, engage graduate students, and there's a combination of both graduate students and undergraduates in those blue jackets in the upper left picture. This is a male elephant seal. We haven't done quite as much work on males. They're harder to anesthetize, and they tend naturally not to come back as well as females. And so we use these tags over and over again. So we tended to do more work on females, but again, it gives you an idea of the size of these animals. What we do know is that males and females do very different things at sea. Here we can see the red dots, these red dots and lines are where females are going. These blue dots are where males are going. And on the right, you can see that the males are going up onto the continental shelf, and this gray is continental shelf, the white is actual land, and the females are staying just off of the continental shelf. So even in the areas where there's a little bit of overlap, they're doing very different things. Why are they doing these different things? Males are much larger than females. And so that larger body size means they have to get more food. So we think part of this has to do with differences in where the prey are most abundant. The other thing we think is that it also has to do that being on the continental shelf is probably a more dangerous place to be, because it's going to be easier for a killer whale or a white shark to get you. But for males, it's really important for them to gain as much weight as possible, because when they come back to the beach to breed, they have to compete with other males, because one male will breed with up to 40 females. And so there's a high stakes game if you're a male. As a female, all you have to do is get the food enough to be able to produce your pup. And there really isn't much competition once you get to the beach. So we think that there's a significant difference in the pressures of what a male does in order to be successful and forage and what a female does. This picture, all these dots are places where the animal went off the air. And you can see the females go off the air much more randomly. The males go off the air in the continental shelf. And we never see, these are animals that not only went off the air, but we never saw them again. So we think that they actually died at sea. And so this is our proxy for where these animals probably were, certainly died, if not were taken by a predator. Now, what we've been able to do is since about 2004, I've been able to continuously put out 20 tags on females on every one of those cycles. And so we now have well over 1,000 tracks. And other colleagues like Roxanne Beltran, who is a new faculty member here, have been able to mine that data set and ask really interesting questions. And this is some work she recently did with that data set that said, well, if you look at where the females are, and each one of these dots is where that female turned around. So this female went way out here beyond the international date line and then came back. This female stayed close and then came back. This one sort of halfway. But what is curious is that they know they all leave here on the bottom. They all leave at about the same time, and they all come back at the same time. And so what this says is that females know where they are in the ocean, and they know when they have to turn around to then come back and make it to the beach. I don't have time to go into it, but it's really to the female's advantage to come back together and hit the beach at about the same time, because then it sets up competition with the males so that the female gets the best male for next year's pup. That's sort of giving you an idea of the overview. What I'm now going to do is switch to, well, how do these animals dive to 600 meters and hold their breath for 20 minutes? And what you think about it, this figure just says, for you and I'm up here talking and I'm breathing. For a seal or a sea lion or any diving mammal, they stop breathing and have to hold their breath. And a friend of mine once said, just think about doing aerobics when you're holding your breath. And that's what any diving, air-breathing diving mammal will do. This is one of the things we do is because we're not big enough market it's very hard for us to develop unique instrumentation. So one of the things we did is take those little heart rate recorders that we used to work out, adapted it for an elephant seal, and you can see here the diving pattern, and this is heart rate of that animal corresponding to the diving pattern. Now what's interesting here is this, at time 80, that something happened where the animal went very deep, and something happened so that its heart rate also dropped. So one of the things we know is that when the heart rate drops, it indicates that there's probably a reduction in the animal's metabolism. It's holding on to the oxygen it has. It's keeping it in particular parts of the body. So we don't know what happened, but it's like we think that the animal may have been responding to being scared by something. If you do these things enough, you can go through and find animals that do really extreme dives. And this is the longest dive that we've ever recorded. This was animal dove for 1 hour and 43 minutes. Can you imagine holding your breath for 1 hour and 43 minutes? What's even more incredible is that the animal did that with a whole series of dives. That this one was 1 hour and 9 minutes. This is 1 hour, 51 minutes, hour 43, hour and 10, almost an hour, with only 2 minutes on the surface. If I had more time, I would tell you about the work that my colleague Jerry Kooiman did, but he was able to show that when animals exceed a certain time of diving, they've used up their oxygen stores and they go into what we call anaerobic metabolism. And when they do that, they typically have to spend more time than they did on the dive, repaying that, what we call oxygen debt. How these animals are able to do that, because there's no question, at least of our understanding of the physiology, 1 hour and 43-minute dive, that animal should have had really high lactic acid levels. So think about when you work out and your muscles burn, that's the lactic acid. So how do these animals not deal with that? Well, they're doing something that we just don't understand. And that's what makes this really exciting, is every time we study these animals, we think we have it figured out and they show us that we're wrong. And usually what they show us that what they're doing was much more interesting. So I said that making custom instruments is difficult. There are a few people who really push the envelope. And this guy right here is Paul Panganis. He was a graduate student here about the same time I was with Ken Norris. He then decided to go into become an MD, got his medical degree at Stanford. And then he secondarily came back to the field. And being an anesthesiologist and an MD, he decided he wanted to really understand this diving physiology. And this is Gina McDonald, who's now a professor at Moss Landing Marine Lab, and Cora Champagne, who's up at University of Washington. What they're doing here is attaching instruments on this animal that will tell us its heart rate and its oxygen, the concentration of the oxygen in its blood. And we release these animals up at, we pick them up on the beach, instrument them, take them to Monterey, let them swim back. Within a few days, they come back. And here's what we see. Here's the diving pattern. So this is a single dive. And here is the blood oxygen level in the venous side. So at the beginning of the dive, you can see that the blood oxygen level increases. That kind of makes sense because there's oxygen in the lungs that are being increasing the pressure increases the oxygen tension. Then as the dive continues, that oxygen level declines. This dotted line is where you or I would black out. And the animal goes down to about 3 millimeters of mercury or 3 torr, which is almost no oxygen left in its blood. And how they do that, we're still trying to figure out. One of the people that took this on was a gal named Jessica Meir, who's right there. This was one of those tracks that shows us the diving pattern in a 3-dimensional space. You can see at the top here, it gets oxygenated and then the oxygen disappears during these dives. The reason I show this picture is Jessica Meir did her PhD at Scripps, but about half of her research was done up here on elephant seals. She then went into the astronaut corps. And if you recall, about 3 years ago, there was 2 women that did the first space flight, the first spacewalk. This was Jess and her colleague, can't remember her name. So Jessica Meir, I used to say, well, we're not doing rocket science, but sometimes it leads to rocket science. And Jess is in the Artemis program. So it's a possibility that Jessica could be the first woman on the moon. And her colleague here, she's scheduled to go on the first Artemis flight. So we do have people that go on to pretty distinguished careers outside of academia, if you will. One of the great things is you get really smart people to work with you. This was some work that a former graduate student postdoc of mine did. He was looking through the diving records and he said, hey, I think we can tell when the female is pregnant or not. And this shows the pattern of an elephant seal. They breed, they go into estrus. They have a period where they have delayed implantation. So the zygote forms, goes into a suspended state. When they come back to molt, the blastocyst implants, and then they go to sea for about 8 months. And during this period of time, the fetus develops and they come ashore and give birth. What you see in the figure to the right is that this red line is non-pregnant females. So these are females that when they came back to the beach, we found they didn't have a pup. And this blue line is females that have a pup. And so you have to think about, okay, this female is producing a pup, a fetus. Well, that little guy, that little gal or guy, that little animal, fetus, has its own oxygen consumption. So as the fetus is growing, it's also demanding oxygen from the mother. And that's what we think is happening here, is that the female's oxygen demands in total increase so that her ability to dive decreases because she's supporting herself and her fetus. Whereas if she doesn't have a pup, mostly what she's doing is gaining fat. So she's not gaining much active tissue. And so she can use more of the oxygen that's stored in her blood for herself. So by just looking at the behavior of the animals, we can say whether or not the female is pregnant. One of the questions when you look at these animals going out to sea for as long as they do, was what are the different dives? And my colleague Bernie LeBoeuf, who started the elephant seal program here at Santa Cruz some time ago, was one of the first to start to think about from a behavioral perspective, well, what do the shapes of these different dives mean? And we looked at these dive records and we figured, okay, the ones where they're going up and down, you can envision a seal moving up and down through the water chasing fish. So we said, okay, those are feeding dives. But then we would get these dives where the animal would just do these very funny patterns. And we put a swim velocity recorder on them. And then we found out that these animals would swim to a point and then just drift. Then they'd come up, surface, go down to a point, and then just drift. So we started thinking about, well, what happens when you have a big meal? You get really tired, right? So you either sit there and relax or you fall asleep. So we call these processing dives because one of the things we see on these elephants is they never spend any time at the surface. For the entire time that the animal's at sea, this is the amount it's spending only 2 to 3 minutes at the surface. To me, that suggests that they're afraid of being at the surface because there are things like killer whales and white sharks that would be an ideal place to find your prey is at the surface. So our idea was that these were the dives where they're possibly sleeping, but probably processing the food after a period of diving. Now, the other cool thing about these so-called processing dives is that when the animal's skinny, since they're sinking, they're floating, I mean, they're just sinking. If they're really thin, they're going to sink rapidly. But as they fatten up, they're going to float or sink slower. And this gave us an ability to start to predict how they're doing in terms of how much food are they gaining, because as this drift pattern changes, it reflects the fact that their body composition is changing. And we can model how much fat the animal's gaining and how that puts together in terms of where actually at sea is it foraging. So we figured those were our foraging dives, and the others were dives where they weren't feeding. One of the other great things about this is we've had some Japanese colleagues who come up with some really interesting technology, and we say, yeah, come on, put on elephant seals. And they developed a little recorder that we put on the lower jaw of the elephant seal, and it would measure the acceleration. We calibrated in the lab whenever an animal would feed. And so we're able to tell that thing would be programmed when it ever had a particular acceleration pattern, that was a feeding event, and it would log when that happened. And that's what these red dots are. Those are every time the animal chased something, opened its mouth in a way that was consistent with feeding. What you can tell is that these dives that we thought were processing dives, the animal's not chasing anything. It's not opening and closing its mouth. You can see up here, that's a processing dives, drift dives, and that all these other dives that we thought were foraging dives are in fact consistent with foraging dives. Well, one of the things from the beginning, we asked ourselves, when do these animals sleep? And whenever I would give this talk at the end, a lot of people would say, when do they sleep? If they're at sea for 9 months and they're constantly diving, how do they sleep? When do they sleep? Well, just in the last few years, I was fortunate enough to get a graduate. Terry Williams and I had a graduate student named Jessica Kendall Barr, who had worked with other colleagues doing sleep studies in fur seals and sea lions. And she said, I wanted to study sleep in elephant seals. And we said, okay. At the same time, a colleague in Switzerland made a little tiny digital EEG recorder. So we figured we'll put these little EEG recorders, and it measures heart rate, EEG, EOG. Jessica made a beautiful little head-mounted neoprene patch, just like you go to a sleep doctor, they put a thing on your head and you go to sleep and they can measure your sleep patterns. Well, Jessie did that. And what we found is that sure enough, during these drift dives is when the animals sleep. And the other thing is when they go down to a point, and then they sort of do these rotations, which are probably just nothing more than the animals completely asleep. We think that the reason that they do this sleep pattern like that is to avoid predators at the surface. We're really excited about this study because Jessie is both a very talented biologist, but she did all these graphics. These are her graphics. This in 2 weeks will be coming out in Science magazine. So we argue that these diving patterns have something to do with predation and avoiding being preyed upon. Again, Roxanne Beltran took the data and said, well, I'm going to look at when these drift dives occur. The idea being is that when the animal leaves the beach, it's really hungry. A female has been there for a month. She's taken a pup from 100 pounds to 300 pounds, all of that from her own stored energy reserves. So she's really hungry. The idea is that maybe they change their behavior while they're at sea, such that when they leave, they're 20% fat. If you're worried about a visual predator, which certainly white sharks are, and to a certain degree killer whales are, you would probably want to avoid diving during sleeping during the day. You want to sleep at night. And in this case, when animals are very lean and hungry and negatively buoyant based on those drift dives, they do tend to do these drift dives after sunrise. After the animal has gotten fatter and accrued the more and more fat, these rest dives are occurring more and more during the nighttime. And so in this period of time, when the animal really wants to spend its time feeding during the best period of time, it pushes these drift dives into the daylight area where we think that these animals are more susceptible to being preyed upon. But if you're hungry and you got to eat in order to survive, you're going to take that chance. Then the question becomes, what do they eat? And one of the problems, I mean, typically what people do is look at the otoliths and the ear bones and the bones of fish or squid. The problem with that is the really fragile bones disappear because they're going through the stomach. And from this, the scats on the beach, we thought that elephant seals ate mostly squid. But we know that out here in the North Pacific, this is where you can see these white tracks are elephant seals. These white lines are tracks of seals. One of my graduate students went out with a Japanese fishing boat, a training boat, and these are areas where they took trawls in the water to see, and they put the trawls at the depth where these animals were feeding. Here's showing the trawl. And this is what the kinds of things you find when you go down to that depth. These guys are called lanternfish. There's these very unusual squid. And what she then did is she collected those fish and squid, looked at the fatty acid profiles. She then compared that to the fatty acid profiles in the blubber and was able to match and show that, in fact, most of the diet of these guys is these little fish, these myctophids who have very fragile inner ear bones. At the same time, our Japanese colleagues now came up with yet another innovation, and that's putting a camera in a tiny housing that would only turn on when the animal was below 200 meters. And they also had it connected to that jaw sensor so that when the animal went to 200 meters and started opening and closing its jaw, it would start taking pictures. And this is an animation of one of those cameras on the head of an elephant seal, somewhere out in the Pacific, 1,000 or so miles at sea, probably in 400 meters of water. We're taking home movies of our elephant seals. And that little thing there is one of those fish that I showed you earlier that the elephant seals are eating. So we know that they're feeding on these deep scattering layer, on those very unusual fish that turns out are very, very common in that range. Now, the other thing that, again, my Japanese colleagues were curious about is they in these movies, you can see here's the diving pattern of the animal. This animal is going very deep, 800 meters or so is a relatively deep dive for a female to go back to over and over again. These animals made fewer capture events the deeper they went, and we figure that means that they were feeding on larger fish, so they didn't need to catch as many. The other thing that we saw in this picture sequence here, you can see the fish isn't moving very far, and on sea down below here, you can see the elephant seal approaching the fish, and the fish isn't trying to escape. There's a thing called the oxygen minimum layer, and many fish go into that to escape other fish predators, but the disadvantage of going there is there's not much oxygen, so you can't move very fast. Well, since elephant seals carry their oxygen on board, the O2 minimum layer doesn't mean anything to them. So it's a perfect place for an elephant seal to go and find prey that is probably lethargic because there isn't much oxygen in that area. This shows that individual. These are more typical dives, but in this one area, this individual was spending a lot of time in that deeper water which we know that in deeper waters where the oxygen minimum layer is. I have a current graduate student that's looking at all of our elephant seal records, trying to figure out how common it is for elephant seals to use this oxygen minimum layer. One of the things as you collect data over time is you see, this is a thing called a Humboldt squid. These things are really big. This is the Monterey Bay Aquarium Research Institute. On this axis, you see hake, which is this species here. This is the abundance of hake that they're seeing with their little ROVs, remotely operated vehicles. This is the El Nino oscillation. And what you really want to see here is these red areas in the figure. These red areas here are observations of these Humboldt squid. And as climate changes, these Humboldt squid are becoming more common in Monterey Bay. And at that period of time, we saw one elephant seal that did something we'd never seen before. That is, she never left the nearshore area. And in fact, she spent almost all of her time. This is Monterey Bay. Moss Landing is about here. We're over here. This animal spent all of her time right there. And what we found out is this is where the local fishermen were getting Humboldt squid. We then looked at her record here in terms of her diving profile and looked at where the Monterey Bay Aquarium Research Institute saw these Humboldt squid, and they matched quite well. We later looked at the blubber of this animal and found that it really was rich in that fatty acids associated with squid. So we figured that this very unusual event was an elephant seal taking advantage of the influx of this Humboldt squid. We also had done some work in Mexico, and we got really surprising records of females that never left Mexico, just like that female here. And I was at a meeting with a number of Mexican colleagues in La Paz, Mexico, and I found out that this area is where the primary breeding habitat of Humboldt squid. So while it was unusual for our elephant seals up here at Año Nuevo to do this, it's probably something that the animals down in Mexico do routinely. One of the things that is an advantage when you do these tags over multiple years is that you can look at what females do the same thing year after year. We had tagged a female in 1995. We normally wouldn't tag older females. One of my graduate students misread the tag, thought it was a 6-year-old. We put a tag on it. Turns out, total serendipity. This female we tagged in 2006 by accident was the same female we put a tag out in 1995. So we have a track of this female as a 6-year-old. We have a track of this female as a 17-year-old. And you can see the tracks look almost identical. I had another student that came and said, I'm going to look at animals over multiple years. And so she put one track after one tag on a female 1 year, the next year. And we found that some females do the same thing year after year. Other females sort of mix it up and do different things. And basically that's as far as she took it. I then had a colleague with a postdoc working with the NOAA lab down in Monterey that looked at this more carefully and looked at all of the females that were either have high site fidelity or low site fidelity. And what we're able to find out is that the females that have high site fidelity go to the same place year after year, are going to a part of the ocean that doesn't change very much. And that females with low site fidelity are females that are sort of mixing it up, going to parts of the ocean that are much more variable. This is chlorophyll, this is sea surface temperature. So the point is that if you look at the variability in the oceanographic measures, these females are doing different things. You then ask, why would they do this? And it turns out we measured the mass gain of these females, and females that did the same thing year after year were doing better, not surprisingly, when conditions were stable. And females that were much more variable were better at dealing with anomalous conditions. And so these sort of different strategies for individuals. So now one of the things we're asking, is there some personalities, is some trait? Are these females that are doing anomalous things risk risk tolerant, and these females risk averse. And that's an area that we're starting to look into. We've also been doing this long enough that we have tracks of mothers. This is the mom in purple and the daughter as an adult female in blue. And the same here is the purple is the mother and the blue is the daughter. So we're starting to try and look at, is there an inherited characteristic to where these guys go? What I didn't tell you is the pup is left on the beach and mom goes to sea. The pup has to figure out how to be an elephant seal on its own. So for them to do the same thing means they're at least following the same cues or have some characteristic that probably came from mother. All this leads to one of the current issues that we face is climate change. And this is some more recent work that graduate student Rachel Holzer did. Some of you remember the warm blob event in the North Pacific where there was a big marine heat wave. Our elephant seals went right through that heat wave. This very dark dotted line is a particular female. This is that track of that female. One of the things we've been doing now is adding a device on the seal that tells us not only where it is, but what the salinity and temperature profiles. Those are things that are really critical for defining the ocean, as well as some of the tags tell us the chlorophyll, which is a measure of the phytoplankton. This is the track of that female as she went through that blob. You can see that the sea surface temperature going down to depth was much higher, the temperature much higher in that region of the blob. And basically you can see that here's the blob. You can see the redder, warmer surface extended through the water column and affected the animal's foraging behavior because the prey actually was going deeper. We were able to compare 1 year to the next. So this was one female during the blob in red, and the following year, we could see that in this case, some females are good at dealing with this. This female in 2015 gained 70.5 kilograms. That was the blob year. She was able to create a pup. The following year, she did a little bit better, 92 kilograms. She also pupped. The 5-year mean for elephant seals is 75. So this female was able to deal with that warm water event, even though she wasn't quite as successful as she might have been, but she was still pretty close to normal. I'm now going to switch gears. I started the talk telling you about Antarctica. I'm going to close the talk by bringing you to some of the work that we've been doing in the Antarctic. This was a picture taken just last year. That's the Nathaniel Palmer in the Antarctic Peninsula. Just some nice pictures of Antarctica. We were there in the winter so that the sun was always on the horizon. Made for quite nice pictures, actually. So this is the area that I'm going to talk about. This is the Western Antarctic Peninsula. So we fly down to Punta Arenas, Chile, get on the ship, and then make our way to the Antarctic Peninsula. In that region, there are Antarctic fur seals, Weddell seals, leopard seals, and elephant seals. And we've been working on all of these species, trying to figure out how they make a living, where they go, what do they do. Taken a lot of our work that we've done on elephant seals up here and taken it down there. The last critter we worked on in this set was the leopard seal, because they got big, mean teeth and they could eat you. The other thing that I'd like to show about this slide is if you look at these animals, there's a common thing in these other seals. I mean, look at the elephant seal, it's got lots of whiskers. The Weddell seal's got big whiskers. Fur seals got whiskers. These guys, the seals, have really big eyes. Fur seals got, if it opened its eyes, it's not as big as these guys, but reasonable eyes. Look at this leopard seal, little tiny whiskers, not very big eyes, a big mouth. Leopard seals feed on fish, feed on almost anything. They feed on fish, krill, other seals, penguins, fur seal pups, fur seals, anything that swims, they can go after. So what we were interested in is how these different species parse up the habitat. How do they use the ocean in different ways? This is a picture of our study site, giving you an idea of the amazing place and the field conditions. the only way we could work on a leopard seal is getting a little help from an anesthetic to keep the animal from biting us. But this shows working where we're taking measurements, gluing these satellite tracking devices on the animal to then see where it goes. You can see a picture of me with the smallest seal, a juvenile female, and the largest female. My graduate student who was working with us at the time decided we had to call them all flower names. So we have begonia and zinnia and this is a male leopard seal. We were able to do 3 of them, but it gives you an idea of the teeth on this guy. That's our satellite tracking device that tells us how deep and how long, as well as where the animal is. When you put all those data together, and these are all the different species now in this figure, the orange are crab-eater seals, the red, the sort of reddish orange are fur seals, these bluish, purplish color are elephant seals, and up here is our Weddell seals. And this time there's not many leopard seals, but you can see the white for the leopard seals. So the first thing is we can see that they're doing very different things in terms of where they go. And this is a little animation that hopefully will work. Yeah, it's going to work. These are the tracks of of all the different species. What you're seeing now are elephant seals. Those orange are fur seals. The purple are Weddell seals, and the white up here, the white are leopard seals. So you can see the patterns are really different. These animals are doing very different things. And we're right at the point of trying to figure that out in terms of how do they partition the environment, how do they use it. One of the things we know is that if you look at dive duration and time, these are crab-eater seals and leopard seals. They're not diving very long and they're not diving very deep. These are mean dives of Weddell seals, and down here we have our elephant seals. And so the animals are both partitioning it in terms of where they go, but also of what parts of the water column they're using. Because these animals are carrying these tags that measure important critical oceanographic parameters, we can start to put the movement of our animals in the context of climate change, of oceanographic features. And we've been putting a lot of these tags out, and back in 2008 is when this region here, this is a satellite image of a place called the Wilkins Ice Shelf. The ice shelf is falling apart, and now the ice shelf has completely collapsed. So this happened in 2008. I was sort of finding any oceanographer who would do something with the data we had. I kept saying, hey, we've got data that during this period of time. A guy named Laurie Padman took the data and was able to show that this is warmer water, this is cooler water. What he's able to show is from our elephant seal records, the water came in this way, went underneath the ice shelf and came out cooler. The reason that's important is when the climatologists figured the heat budget for this ice shelf, there wasn't enough heat coming from the atmosphere for it to collapse. But when you added this warmer oceanographic water coming in and eroding underneath that ice shelf, that closed the budget and allowed them to understand what was happening. So the last beat of the story is comparing different species again. Now this is, the white here is ice, the blue is open ocean, and this green is open ocean but warmer water. The red dots are elephant seals, the green dots are crab-eater seals. These are crab-eater seals on the right, elephant seals on the left. The thing I want you to see is that the elephant seals are moving in and out of the ice and into the open water. The crab-eater seals are staying in the ice. So it wasn't really a surprise. So what we expected is that where you see crab-eater seals is in the ice. That makes us concerned is what's going to happen to the habitat of crab-eater seals as the climate changes. And so some colleagues at Old Dominion University, this is the ice conditions today. When they run their models of how the climate is going to change, you see that the distribution of sea ice is much sparser. This is current conditions. This is future conditions. And in fact, this is what we're already seeing. So these models are doing a very good job of predicting. We have a cruise next month, May and June, to put tags on crab eater seals. And what we're already finding is we're going to have trouble on that cruise because the ice conditions are increasingly poor. But why do we care about ice? Well, ice is the primary habitat for this little critter called krill. It's a little shrimp-like critter, but krill is the major prey item of crab eater seals. And if you look at the teeth of crab eater seals, you can see they're really unusual, and they act as a sieve to allow this crab eater seal to eat the krill. So we went on 3 different trips and studied these crab eater seals, putting these time depth recorders out. You can see the records, the tracks. My graduate student, Luis Huckstadt, put all these data together, predicted what the habitat of these animals would be like as it changes. And this basically shows how the habitat will change in the Antarctic. And there'll be an increase or loss of habitat. That's what our current cruise is, to put more tags out on. These data were taken in some cases 20 years ago. So we're trying to see how things are changing. The last thing I want to talk about is not doing this in isolation. There's a number of different investigators that are putting tags out on seals, on penguins, on albatross, on whales. And one of the great things about working in the Antarctic is you go to meetings where all these people who work in Antarctica come together. And at one of the meetings we said, let's put all our data together. So those are all the people, and here's all the data. We now took all of these species that you see here, pulled all these data together, put this animation together, and then said, where are the biggest habitats? Where are the most important areas of the Antarctic Ocean, the Southern Ocean, for air-breathing vertebrates, mammals and birds? And we found these regions that you can see highlighted in that orange red. We then defined areas of ecological significance and asked how many of those areas are currently protected under marine protected areas. And what we were surprised is that 27% of the region is currently, areas of ecological significance are currently protected, and proposed marine protected areas as much as 39%. And this is, if you took all of Antarctica, it's a much lower number. But the point is we're protecting the right places. We took that a step further and said, well, how is that going to change? We know from the models of climate change how the oceanography is going to change, but those marine protected areas are static. So we asked the question, here's the protected areas, the habitat is going to move. How does that affect the different regions? And what you can see is that there are some places that will get better, and there's some places that will get worse. So that's sort of my slide rules to satellite tags presentation, and I'll answer any questions if there's still time.
Okay, we're a little tight on time, but we can have a couple of questions here. Todd.
Interested in the timing of when the elephants will all turn around.
No, they turn around depending upon how far they are. So a female that goes much further offshore turns around sooner than females that just go a little ways offshore. So the ones that just go a little ways stay out there longer and then turn around. So they all turn around at different times, but they all come back. They all arrive at the beach at the same time.
Thank you. Garrett, you have
How do elephant seals solve the nitrogen problem, which he's referring to as decompression sickness? We used to say that it's because when they dive on an exhalation, so there's no air in their lungs, and so that reduces the amount of nitrogen that'll go into the lungs. People have been studying this more recently, and if you do many dives one after another, you can build up enough nitrogen in your tissues, especially if you're making 20-minute dives with 2-minute surface intervals. You don't have enough time on the surface to download that nitrogen. We don't know. Are they affected by what?
Well, the garbage patch.
Oh. The garbage patch is a little bit further south. I mean, there's garbage everywhere, but the big garbage patch is in the Central Pacific Gyre, which is further south than where these guys are feeding. So I don't know the answer, but I don't think so because geographically, it doesn't mix. So you asked how long the sensors last? We can put a tag on it. It won't last longer than a year because the elephant seal molts and we glue the tag to its hair. So it'll never last more than a year. Much of the data are available in real time. All of the oceanographic data are transmitted through the global telecommunication system and are absorbed into the world oceanographic climatology. Because the funding for the satellite data uplink is funded by NOAA. The real-time data of where these animals are is available on the animal tracking network. So you could go online and see whoever's out there, there'll be these data. We are currently, all of the diving and tracking data and all of the data that I showed you, where there's a lot of data, not all of it's available. We're in the process of putting together a massive data set that we're going to publish so that anybody could do all the stuff I just did with it. So that's, or in a few months, that'll hopefully be out. Great question. And that's what Roxanne Beltran is currently studying. She got a grant from the National Science Foundation the title of the grant is Rules of Death, trying to figure out, looking at pups on the beach, taking a whole bunch of measurements, putting satellite tracking devices on, knowing that half of them won't come back because at least half the pups we don't see again. So she got the funding to put enough tags out that we can start to answer that question.
Okay, we're gonna have
Link.
Together, but I don't but in C, it looks like
Yeah, they're on land, they're gregarious. There is a social system where the males identify each other and they know each other because former student played back acoustic calls of different males and they recognize the alpha male from the subordinate males, and they will either attack the speaker if they're an alpha male or run away from it if it's a subordinate. The females really probably don't interact much other than they're all together. And at sea, they're so far apart. But I have a colleague that asked that question. We gave him all he's had a bunch of computer guys that analyze the data, and the animals seem to move in synchrony. And so he argued that it was sociality. But the problem is that the animals are hundreds of kilometers apart. So A, how would they communicate? And B, what would they talk about? You know, if you're 100 kilometers away from your buddy, what good is, you know, what are they going to say? So I think it's more of a artifact that they're all doing the same thing in terms of the timing becomes very important. So they all return to the beach together.
So if you have questions, maybe Dan can hang around for a little
Sure.
bit. But terrific, Dan.
And before you go, please, to help everybody, if you could look around at the play and make sure you're not leaving any cups and papers and all that stuff to take care to one of the cans. And if a few of you are able to help restore the space to the cafeteria, that will help the students who have been here serving us. So thanks very much, everybody.